So let us start. So very good morning to all the participants. Welcome to you all in today's international webinar on genomics and cancer therapy. Now the most dreaded disease, cancer, and gene are found to be inseparable today. Certain aberration of the gene is causing the cancer. Today, we have with us a scientist who is by profession is searching the genes relating to cancer and providing extremely valuable information of oncogenes to the doctors, suggesting a suitable therapy for a certain patient, and again keeping track with the data that is generated from those therapy for future research. Let us welcome our speaker today, Dr. Sunny Guin. He is the principal scientist in Translational Research, Bioinformatics R&D, Semaphore, which is a health intelligence company, presently situated at Stamford City, USA. Achha, everybody can listen me? Hello?
Yes, sir.
Okay. So he is Dr. Gui. He is a B.Pharm from our own university, when its name was West Bengal University of Technology. He achieved his Ph.D. in pharmaceutical science from Graduate School of Biomedical Sciences, Texas Tech University, Health Sciences Center, USA, under Professor Ming Hai Wang. He is trained in cancer biology and therapeutics as a postdoctoral fellow at Department of Surgery and Urology, School of Medicine, Surgery and Urology, School of Medicine, University of Colorado, USA. Now he acted as research instructor in University of Colorado. So he is trained in cancer biology and therapeutics as a postdoctoral fellow at Department of Surgery and Urology, School of Medicine, University of Colorado, USA. He acted as research instructor in University of Colorado, and he acted as research scientist in Cabra Cancer Research Institute. Next, he acted as senior scientist in Bioinformatics R&D Semaphore, and he's actually acting there presently in the as a principal scientist, and he's acting in the same company. He has published 19 international scientific papers in reputed international journals. All are peer reviewed. He was awarded the BCAN, ECAN, this your investigator award. Now BCAN stands for Bladder Cancer Advocacy Network. He was the bronze medalist of our university in B.Pharm in his year. He has been awarded with 4 research grants till now. Therefore, we have with us a learned man. We may call him an authority in genomics and cancer therapy. Sir, now I wish to hand over the session to you. Now, before leaving, all the participants are requested to go to our website, nscbip.org, and click on the feedback form, fill it up with correct Gmail address after this session ends. It will remain open for 1 hour. After that, your certificates will be shared with your Google Drive. Mind it, not in email address. In your Google Drive, you will find it shared. So you will find your e-certificates in the shared with me folder of your Google Drive. So thank you all. Now over to Dr. Sunny Guin, sir.
Thank you, Dr. Samantha. Thank you for the very kind introduction. I will share my screen. So I will be mainly talking today about, so everybody can hear me and see my screen, right? Okay, so I'll be talking about genomics and cancer therapy. I'll be giving a brief introduction, or the presentation pretty much summarizes what's going on in the realm of cancer therapy currently in terms of genomics and targeted treatment. So Dr. Samantha already gave an introduction. I graduated with a Bachelor of Pharmacy from Gupta College of Technological Sciences. I was part of the first batch coming out of the university. I did my PhD in Texas Tech Health Sciences Center, Graduate School of Biomedical Sciences. My main dissertation was targeted drug delivery, targeting a particular receptor tyrosine kinase. After that, I did my postdoctoral training in University of Colorado Anschutz Medical Campus. Currently, I'm a principal scientist at Semaphore, which is a big data genomics company. Before that, I was a research scientist at Kabara Cancer Research Institute, where I had my own lab. And I was driving research projects. Before that, I was a research instructor at University of Colorado. So I'll be starting with the concept, which I think most of the students are familiar with, central dogma. So from DNA, RNA is made, and eventually it leads to protein. You know, we all know parts of DNA which can code a protein is called a gene. So, you know, the DNA makes RNA and leads to protein. And this forms the basis of all our research. So after introducing the central dogma, I would like to introduce how cancer progresses. Basically, you know, you have your normal cells. You might have a genetic alteration in one of these cells, which leads to rapidly dividing cells, which is initially called hyperplasia. It's not a cancer at this stage. You later have dysplasia, where the cells change form. Then you have in situ cancer, where the cells are growing rapidly, but they're not like going to other parts of the body. They're still restricted to that particular site. But then eventually you have malignant tumors, where the cells are aggressively growing, is invading other tissues, growing into the bloodstream and metastasizing to different sites. So these are the different stages, and it starts off in most of the cases with genetic alterations. And this genetic alterations could be due to various reasons, from toxic reagents like smoke, environmental issues, and could be in some cases also germline genetic issues, where you have certain mutations which comes from generation to generation, and that leads to cancer. So here I want to show what are the major factors contributing to cancer. So for a cancer cell or a particular cell to grow rapidly, you have to have a sustained proliferative signaling. So there should be proteins in the cells which have, due to activation of certain proteins, they keep growing at a faster rate. And it is able to suppress functions which slows down growth. Besides that, you have a metastasis, so proteins or genes will drive these cancer cells to get out from that specific organ site and go to a different organ site. So, you know, proteins which enable that process. Also, we are probably familiar with TERT proteins. So TERT helps in replication. So if you have mutation in the TERT gene, you can have continuous replication, so which reduces the mortality of the cell. So a particular cell can live for a longer period of time. Besides this, there are signaling which inhibits cell death and induces angiogenesis. Angiogenesis is increased blood flow to the tissues. So you can have protein alterations which will increase the blood flow to the tissues, which will enable oxygen and nutrients to go to these cancer cells, and leading to rapid growth. So these are the main factors contributing. And all of these are predominantly controlled by two sets of genes. One is called oncogene, one is called tumor suppressor genes. So oncogenes get activated, and activation of these genes lead to, like, it's a gain of function. It turns on these genes, and their activity is increased, which drives to tumor growth. And then there is tumor suppressor genes. So these are genes which keep the tumors in check, or a cell growth in check, when their function is lost, then the tumors grow at a much faster or a rapid rate. To give, to explain or show how gene and oncogene and tumor suppressor genes work, you can think of them like a brake. So tumor suppressor is a brake in a car, whereas an oncogene or a proto-oncogene is an accelerator. So the brake prevents the growth of cells, whereas accelerator, in a car, it helps in acceleration. So in what happens in cancer cells when these genes get mutated, it's like the brake of a car is broken, so it cannot stop, and the car is going fast. And oncogene is somebody has pressed on the accelerator and not releasing it. So again, the car is going fast. So when you have both of these genes regulated due to mutation, so you have loss of function for the tumor suppressor genes. So it cannot inhibit the growth, whereas oncogene is continuously activated, which is accelerating growth. You have rapid cancer growth. So having introduced the concept of oncogene and tumor suppressor gene, I would like to focus on receptor tyrosine kinase. So, students who have taken pharmacology must be familiar with receptor tyrosine kinase. These are a type of transmembrane proteins which have signal transduction. When there is a ligand which binds to these proteins get activated and have downstream signaling. So these receptor tyrosine kinases are a very well-known, a lot of these receptor tyrosine kinases are well-known oncogenes. When these receptor tyrosine kinases get activated due to mutation or overexpression, you have signaling through them which induces proliferation and suppresses the death signal, which results in rapid cancer growth. So an example of a receptor tyrosine kinase is HER2. It is very well studied in the breast cancer field. The protein is called HER2, the gene is called ERB2. So what happens in a certain fraction of breast cancer patients, you have this receptor tyrosine kinase overexpressed. So this is a cartoon which shows a normal cell which expresses a few copies of HER2 gene or the HER2 protein, whereas a fraction of breast cancer cells express a lot of these protein and have rapid signaling, downstream signaling through HER2, which results in growth of these cancer cells. This is just showing how HER2 expression looks at the protein level in breast cancer tissues. So this is a picture from IHC or immunohistochemical staining. This is a particular type of antibody-based staining which is used in clinic to detect expression of this protein. So this is an example of a breast cancer tissue. The brown color represents the expression of the HER2 protein by immunohistochemistry. So a positive cell, a breast cancer tissue which have positive HER2 expression, you see a lot of brown staining, whereas a negative cell, you see there is no staining. So this is how the physicians or the pathologists distinguish between positive HER2 cell and negative HER2 cancer. So what happened when scientists started discovering these oncogenes, these receptor tyrosine kinase, they thought, okay, these proteins are getting discovered. They seem to be a major driver for these cancers. So can we build a drug targeting these proteins? So trastuzumab, or the brand name is Herceptin, is the first targeted cancer drug, which came out, I think, in 1998. This is a monoclonal antibody. Again, students who have taken immunology should be familiar with antibodies. So this is an antibody-based drug. What it does is when the cells are overexpressing HER2, it binds to the HER2 protein and inhibits its function. So downstream, you have inhibition of proliferation and antibody dependence cellular cytotoxicity. So this is how the antibody, targeted drug is able to kill cancer cells which express these receptor tyrosine kinase. So once scientists started discovering these receptor tyrosine kinase and started establishing their roles in cancer in the late 1990s, it became a very hot topic in the field of drug discovery, because people were thinking, now you discover these proteins, you target them, and you can kill these cancer cells. So this is a timeline of trastuzumab. So RB2, the gene, again, the gene for HER2. HER2 is the protein. The gene is called RB2. It was discovered sometime in 1980s in breast cancer. And then around 1998, trastuzumab was the first targeted agent discovered for treating HER2 positive breast cancer cells. And you see, as time progressed, there are more drugs which have been approved over time for HER2. So people have started, they make one drug, then they start making another drug, with varying levels of toxicity to treat these patients. So since 1998, when trastuzumab was discovered, it was, as I said, it was a very hot topic. So there was a lot of study going into receptor tyrosine kinase and their role in cancer, and trying to target them. So since then, you can see I have listed all these different families of receptor tyrosine kinase, which scientists started studying and tried to make drug against. And I wanted to, actually the circle has gone on the wrong gene. So when I went for my PhD to United States, I was working, my dissertation was on one of these receptor tyrosine kinase. It's actually the RON receptor tyrosine kinase. And my main thesis was to develop targeted formulation against these receptor tyrosine kinase. In 2006, when this was really a big topic. So this is just to break from the science. I have put a picture of the campus. This was the Texas Tech Health Sciences Center campus in Amarillo, where I was a grad student. So this is the medical campus. So here you see this was the School of Medicine. The building in the center, the building on the right, this is the School of Pharmacy. So this held all the undergraduate Bachelor of Pharmacy classes, had some labs. This building here, the two-story building here, was the labs for the PharmD students. So, Bachelor of Pharmacy students, all their clinical and pharmaceutical labs were all in this building. And this two-story building on the left was the graduate labs. So all the PhD students, all the PhD program labs were situated in this building. So this is where we were. It was a pretty nice building, has a nice cafe in front, and then had really great facilities. And this is part of the campus. On the other side of the street, there was the university hospital, library, another couple of administrative and research buildings. So going back to 2006, when I went for my PhD, I was working on this RON receptor tyrosine kinase. It is a transmembrane receptor kinase. It binds to this ligand called MSP, macrophage stimulating protein, which induces downstream signaling. So my work, what my lab used to do is build monoclonal antibodies against this protein. And my thesis was to build these immunoliposomes. So again, I think in pharmaceutics, you probably have learned about liposomes. The students have learned about liposomes and immunoliposomes. So liposomes are lipid-based formulations, and you have a drug entrapped in it. And our lab used to make antibodies against this receptor tyrosine kinase. So my project was to make liposomes, optimize them with the drug uptake, and then attach antibodies to them and then target cancer cells, which was overexpressing these RON receptor. So this is just a cartoon showing we make this formulation, we target these tumor cells. We used to look at uptake, how much of these liposomes would bind to these receptor and was taken up in the cells, and then looked at cell death. So doing the in vitro model, and then in the mouse model, I used to test how these formulations were working. And actually it did pretty good. It was taken up by a pharmaceutical company to develop further, but it kind of got lost after that. But initially we had really good progress on this and trying to take it to the drug development stage. So from there, I have introduced the concept of oncogenes, receptor tyrosine kinases, targeted drugs. So I just want to, at this stage, give a snapshot. A physician, when a cancer patient comes in to a doctor, what are the different treatment options a doctor has for their patient? So they can have hormone therapy, which is very relevant in breast cancer, other woman-related cancers such as uterine cancer, or in male cancers like prostate cancer. So they have very specific hormone therapy. You have surgery as a treatment option, bone marrow transplant for basically blood cancers. You have chemotherapy. I think pharmacology, you have learned about chemotherapy, which are DNA intercalating agents like Taxols and platinum agents and all these chemotherapy, which have a broad function. Targeted agent, targeted therapy, which basically function by inhibiting a protein function, very specific the way they act. Radiation therapy, this is also very general. You have a tumor, you just radiate that tumor and kill those cancer cells. And immunotherapy, this is another very new topic in the last 4 or 5 years. I'm not going to cover it today. So what I want to say is a physician, when a patient comes in, has a lot of treatment options. So how do the physician make a proper informed decision on what is the best treatment for a patient? So this brings us to the concept of precision oncology or targeted therapy. So what is going on is currently the physicians and the scientists are trying to come up with the approach of molecular profiling of patients. The molecular profiling could be a protein-based like IHC, as I shown in one of the earlier slides for the HER2 protein. It could be genomic-based, where you're looking at doing sequencing and looking at mutations in patient samples. So once you sequence or look at protein biomarkers, you can probably break the patient population down into various subgroups. And these subgroups can have, based off prognostic markers. So prognostic markers will tell how a patient will do on a treatment. Or will they have favorable response, or they will do bad? Markers which predict sensitivity to drug or resistance. So you have certain mutation, or you see by protein expression, you see certain proteins which will tell you, okay, these patients are not going to be responding to a particular drug or going to be respond very well. Or markers which predict adverse events. So sometimes you can have certain markers which will say, okay, this patient is not going to metabolize this drug properly, and so won't respond. So, if you do a molecular profiling for patients, you can probably identify patients who will benefit from a particular treatment or won't benefit from them. And you can have better informed treatment for these patients. Again, sticking to the example of breast cancer at this stage. So if you have 10 patients from breast cancer, so breast cancer has been very well studied. So 6 out of 10 breast cancer patients will respond to hormone therapy. So these breast cancer patients generally will have overexpression of estrogen or progesterone receptor and will do very well with hormone therapy. And these patients will have eventually slow growing tumor. So 20% of these patients, of breast cancer patients, express the HER2 receptor, which I showed earlier. And they will respond very well to trastuzumab or similar drugs which inhibit HER2. that leaves you with another 20% patient who are negative for estrogen, progesterone, or HER2, and these patients do well on chemotherapy because you cannot give them any targeted or hormone therapy. So this is how, if you do a profiling with whether it's protein-based, you can segregate patients and show that identify patients who will respond better to a particular drug. So this is actually, again, to take a break from the concept. This is a picture of the University of Colorado Anschutz Medical Campus, where I was a postdoctoral fellow and a research instructor. This is the whole medical campus. If you see the building here, this is the main hospital. This building here is the children's hospital. The children's hospital in this campus was ranked #3 in the country. It's one of the best children's treatment center you can find in United States. And as scientists, we were holding in these buildings. So you see there are 3 buildings here, 1, 2, and 3. These 3 were the main research buildings. So this one was RC1 North, this one was RC1 South, and this building, or research center and research complex, and this building here was RC2. So this is where my lab was. And you can see from this building, you can look at the background. You have a very good background of mountains, the Rocky Mountains in Colorado. You could see these mountains from the lab. And here at the background, you see this is the Denver, which is the capital of Colorado, the main town center, or the downtown. So this was a very good campus, very scenic. I spent 5 years here, and I spent it. The reason I'm showing it here because I spent a lot of time doing trying to identify patient cohorts who might respond to a particular type of treatment in bladder cancer. I'm not showing those examples, but I worked in a urology lab, and my main focus was in bladder cancer. So from there, so now I've introduced the concept of oncogenes, targeted therapy, personalized medicine. So I want to next move on to sequencing. So when people started understanding that you have to have, you need to identify proper markers to really move forward the field of precision or personalized medicine, there was a big focus on sequencing to identify mutations in patients, because you can do only a limited amount with protein and protein-based biomarker studies. You can only look at a few proteins. There are a lot of limitations to do a high throughput protein screen across various proteins. If you can improve your genomic sequencing capacity and technology, you can take the DNA from a tumor and you can sequence it, and you can identify hundreds or thousands of mutations which might be relevant in cancer, and you can better segregate patients and understand how they might respond to a particular drug or help with drug development. So in around 2001, sequencing had started, but it was a very expensive process. So this is a graph showing when sequencing really started in the United States, sequencing tumor samples or genomes. So in 2001, the sequencing to identify mutations started, and you can see the cost per genome at that time was $100 million per genome, which is, you cannot provide this to patients because it's going to be very expensive. But with advance of technology, you can see around 2008, there has been a significant drop at the price of sequencing. And around 2015, in 15 years, what used to call $100 million now costs $1,000. So this is how the sequencing has developed in a very short period of time, and the price has gone down. And this has enabled enabled cancer centers or genomic companies to sequence patients in big numbers because the cost has gone down. It is affordable for people to do. So I will show you what advantage sequencing has. So again, going back to the breast cancer example, so I have introduced HER2 overexpression in breast cancer, right? And I have shown this by this IHC showing breast cancer patients by protein have high HER2 expression. And these patients are treated with, the first drug approved was trastuzumab, but now there is a list of a bunch of other drugs, lapatinib, neratinib, adotrastuzumab, panitumumab, like many drugs approved for HER2. So here in this graph here, the ones with the red lines, I'm showing the mutation in this gene. So the gene for HER2 is RB2. This type of graph is called an oncoprint. So how to interpret is each bar here represents one patient. So you can see by each bar, it is one patient. The red means amplification. So amplification of the gene leads to overexpression of the protein. So what was happening earlier, the patients who had this amplification would have the overexpression and it was being detected by IHC. But as you can see, there is a fraction of patient, the green dots here represent mutation. So there is a fraction of patient who have mutation in these gene, in RB2 gene. So these patients were not detected by protein, by the IHC protein expression, because IHC cannot detect mutation. Only sequencing can detect mutation. So there was a fraction of patients who are getting missed, who would respond to these drugs, which was approved for HER2. The reason being they were not getting sequenced. So once this large scale sequencing became possible, people started identifying these mutations in RB2 gene, which was activating and was having a similar effect as overexpression of the gene has. And this is an example below here from a patient study. So there's this breast cancer patient who was RB2 negative, so did not have overexpression of the gene, but have a mutation in the RB2 gene. And this patient actually responded to neratinib, which is one of the HER2 inhibitors. So you can see this is a plot which shows the progression of the disease. So the patient was put on neratinib, had an initial response, and then when neratinib was combined with a chemotherapy, had an even stronger response. So this demonstrates, and this graph here on the right shows that actually for HER2 mutation across different cancer types, you see a response with this drug, neratinib. So this points to the concept that when you start identifying mutations, you can find a larger fraction of patients who will benefit from this targeted therapy. So that's the point I'm trying to make. So when you had only IHC, you are detecting a fraction, but you are missing patients. With mutation, with sequencing, you are able to find more patients who can respond to these drugs. So this is just a snapshot. I don't want to go too much here, but with large sequencing study, you can look at mutations across several genes in a particular patient. This is just showing a bunch of different genes. Again, one bar here represents one patient. So if you go down here like this, you can see this patient, this first patient had a mutation in amplification and mutation in HER2, and in EGFR and in a bunch of other genes. So you can look at a lot of genes at the same time, and this helps you interpret how a patient might do on a particular treatment. Also on a gene level, you can look at a mutation. So this is called a Lollipop plot. and you can look at in a particular gene where these specific mutations lie and can quickly identify which amino acid could be relevant. So you see this is a KRAS gene, and you see that at this G12 or glycine 12 site, you have a lot of mutations. This type of analysis also helps you identify which mutations might be relevant. If you look at these frequencies, you start seeing that, okay, this mutation is happening a lot, so it probably has more functional relevance. So I was involved in a lot of these sequencing studies when I was working at Gunderson Health System or Kabara Cancer Research Institute. I used to work with a lot of oncologists and clinicians, assisting them with treatment of patients. So we used to work with them, interpreting molecular data and help them make informed decision for patient care. This is the main hospital, snapshot of the main hospital of there. So, working in a hospital system, I started on getting more and more into precision medicine. And I thought, just looking at these mutations and asking the physicians, what might be the best treatment option, or advising them is the least we can do. There is more we can do if we can combine genomics as well as outcome data. So that is why I moved to a big data genomics company who provides sequencing service to patient, but also collect patient information and combine that to come up with better treatment strategy for these patients. So this is what we do at Semaphore. So we provide genomic sequencing to patients. So now a physician, when they think a patient might benefit from sequencing and targeted treatment, they can send samples to our company. We will provide the information back to them, saying what mutation a patient may have, what type of clinical trials or drugs might be available for the patient. Also, what we do is obtain all the clinical data from electronic medical records. So in the States, there is a very good system of the patient records being maintained in hospital system in a very structured way. So a patient, for the first day they were seen in a hospital, all their clinical data is maintained in a structured manner. So we get all those information from hospital. And we try to combine those two and analyze the data to come up with better treatment strategy for patients. We also do, when we identify some novel mutations in patients or new mutations, we also build research projects where we characterize these mutations. So we'll make these mutations in a cell line or in a mouse model and see how it impacts tumor growth. So combining all of these three approaches, we can come up with a better treatment strategy for patients. So this is what was missing in my earlier hospital system, where we mainly focused on this first part, and we never got a detailed insight because we are not able to combine the other components. So that's what was very unique about a company like Semaphore, and that's why I moved here. So I will be showing briefly the clinical workflow. So when a patient has a tumor, the physician, the pathologist, they will get a sample of the tumor, we will sequence it. It goes through a bioinformatic pipeline which identified what mutations there are. So once the mutations are identified, there has to be an interpretation of that information. So what does that mutation mean clinically, right? Whether it's activating, are there drugs available? So our company, and this is where I was initially involved, have a cancer knowledge base. So the knowledge base contains information on several mutations. So these knowledge is collected from published literature and other various data sources. And these information then is translated into a report, which is then provided to a physician who helps make a treatment decision based on the information we provide. So what does the cancer knowledge base contain? So it contains state-of-the-art knowledge of all the mutations, thousands and thousands of mutations, which is known across hundreds of genes. What do they mean? in terms of function, what do they mean in terms of prognosis, treatment, clinical trial, we have all information. What type of treatment information, guideline, clinical trials, I already mentioned that. And also we want to build a system how this information can be quickly transferred to the reporting stage. So, you cannot maintain this information. You also have to build a high throughput system where this information can be transferred quickly to be put in reports. So we also work on that. So, I mentioned about reports we provide to the physician. So this is a sample report. So you can see how a sample report, which we provide to the physicians, there is an interpretation section, which summarizes the finding. Here you'll have the genes. So this patient had a mutation in the KRAS gene and ATM gene. We provide information if there are drugs available, approved drugs available for this gene, or if there are contraindicated drugs. So as you can see for KRAS, there is contraindicated. So these 4 drugs are contraindicated. That means if the patient comes up with this mutation, the doctor should not be recommending these drugs. We also provide clinical trials. So if you go back to the previous page, you see the patient has a KRAS G12C mutation. And what are the clinical trials available for that specific mutation? So we list all those clinical trials so that the physician can make an informed decision if the patient qualifies for any of these clinical trials. We also provide here a description of what this mutation means. So this is an example of KRAS, what this mutation means in terms of prognosis, therapy, what is the frequency of this mutation in this cancer type, similarly for the other gene. So this is how a detailed report is put forward and sent to the physicians. So that brings us to the next step. So we send this report and we have the EMR reports from the physicians. So what we want to do is we are sending these reports and we want to see how the physicians are treating these patients, right? So this is an example. This is a very simple analysis. So, we have listed or we have seen what are the different targeted agents the physicians has recommended cancer patients. And this is a lung cancer patient cohort. So we can see all these erlotinib, afatinib, osimertinib. These are all targeted agents the patients were prescribed. And number of cases demonstrate the number of patients. And biomarker gene level. So for erlotinib, EGFR is a biomarker. So if the patient has an EGFR mutation, then the doctor will prescribe erlotinib. So what we can see here is whenever a patient has been prescribed erlotinib, there is always an EGFR mutation. So we can see from here that the physicians are following the standard treatment. There is a lot more deeper retrospective analysis we can do with these in terms of patient outcome, but that's for another topic, another day. But what I'm showing here, when we have the outcome data for these patients, we can clearly see if the patients are being treated of the specific biomarker. Here is the column demonstrates FDA approved or NCCN guideline. So the drugs here, are they approved or are in the NCCN guideline for lung cancer? So if Y means yes. So we also see a couple of interesting cases where there is this drug combination here, which is not in the guidelines, but was used by the physician. So this is demonstrate off-label use. So these are very interesting things where we can follow up on these patients and see when the physician is giving some drug off-label, how the patients are responding to them. We can also look at when a patient is moved from one drug to another, what is the reason for the drug change? So you can see when erlotinib, osimertinib, these are all EGFR inhibitors. So EGFR is another receptor tyrosine kinase. Erlotinib is a specific drug against it. Osimertinib is a specific drug against it. So when the physician is changing one drug to another, what is the main reason? Sometimes there are resistant mutation, sometimes there is dose-related toxicity, sometimes just the tumor progresses and they put on another drug. So these type of analysis helps the physician understand what are the shortcomings of certain drug. So for example, 5 patients have been moved from erlotinib to osimertinib due to dose-related toxicity. So when the physician sees this, they see that there is significant amount of toxicity related with this drug. So next time they might just start with osimertinib and then they won't choose this drug to start with. So when you show them these type of data, they will able to make a more informed decision saying that, okay, we don't want to start with erlotinib. It results in toxicity in a high number of patients. We'll start with the other drug instead. So when we give this feedback to them, it helps with the patient care. And I also want to say, so our company also builds software tools. So, you know, we do sequencing, we have the EMR data from the patient, but is there a simple way to demonstrate the data so that a physician or a patient can quickly look at their journey? So this is a case study. A patient who had an EGFR mutation was moved from erlotinib to osimertinib. And this is how it looks in the software our company builds. So you can look at different time points and see at what time point sequencing of the tumor was done. And based on the sequence, when the metastasis of the tumor happened, so the tumor was initially restricted to the lung in this case, then it has a metastasis at a later date, when the targeted treatment with erlotinib started, if they had a sequencing later, when was the second sequencing done, if the drug was changed, what was the finding for the from the sequencing, if the drug was changed, what was that, and if another sequencing was done, what were the findings from that, and you know how the treatment proceeded. So you can at a snapshot look at all this data in your screen instead of going through thousands of pages in in notes in a computer screen. So you can just open up this patient, you type in the patient ID, and you can just in a quick snapshot see when the mutation testing was done, what are the different drugs given, when was the drug changed, and the physician can quickly understand what is going on with the patient instead of going through pages and pages of report. So this is what as a company we are trying to achieve, to make the patient care more easy and informed. So that's where I'll finish. This is a picture of the corporate office of Semaphore. So not all of these buildings, like 4 floors in this building here is where our office is. Besides this, we have offices in New York City. We have offices and labs in a couple of other small towns, but this is where our corporate office is. It's just by a Bay Area. It opens up to the ocean, Atlantic Ocean, in half a mile. So it's very pretty. You can take walks in afternoon or in the evening outside the building. It's really nice. So that's where I will finish, and I'll take any questions.
Thank you, sir. Hello. So such a informed delivery of lectures you have given. So if anybody has any questions, please let me know. Hello to any participants. Hello, can you hear me?
Yes, sir.
Yes. So any question? Okay, now actually I have one question. Sir,
Yes.
Is the genome approach to cancer therapy is presently employed in India? Have you any information?
I think it is employed, but I don't think the scale is as big as it is in United States or in other bigger European countries. I think in India you have more smaller panel tests where they just test for a few genes. Like in breast cancer, they will check for HER2. They might check for HER2 mutation. In lung cancer, they will check for EGFR, maybe through single gene test. They will check for ALK, ROS1. They'll check for probably 5-6 genes, but not through a multi-gene sequencing panel. They will just probably do single gene test, unless maybe in AIMS or in some big institutes or Tata Cancer Center, they might have some a little bigger facility where they can look into a little bigger group. I think that's the real scenario in India.
Oh. Now, what will the probable cost or approximate cost? I'm not asking.
The cost varies. So in United States, medical cost is pretty high. Whenever you go through a hospital system and for the sequencing, it might be, if you look at the raw cost, it might be like $4,000 to $5,000, but a lot of it is covered through insurance. So the patient might out of pocket be maybe a couple of $100 to $300. So it is expensive, but mostly it is covered. Also, big hospital systems have very good philanthropy and support systems. So if the patient a lot of time are not able to cover the cost, the hospital bears the cost for this sequencing. Because there is a lot of interest in research, so there will be funding available to oncologists, and they will run the sequencing through their research fund and get it done, even if it's not approved through insurance or if the patient cannot pay for it. So there is always options.
Okay, let me see whether in YouTube their questions are there. One question is there, sir, by some Rishabh probably. In case of breast cancer, which is more suitable and most working hormonal therapy or targeted treatment, which is more preferred?
So it depends on the type of tumor. So breast cancer can be broken down into three broader categories. So we have your estrogen receptor positive, progesterone receptor positive cancer subset. You have your HER2 subset, which has high expression of the HER2. And then you have triple negative breast cancer, which don't have the hormone receptor overexpressed or HER2 overexpressed. And these three subtypes have completely different treatment strategy. So the HER2 positive and the triple negative won't benefit from hormone therapy, but the estrogen positive and the progesterone positive will respond to hormone therapy. So the question is, which is prevalent? More prevalent is both are equally important. You have to identify the patient who will respond to that particular treatment.
Okay, thank you, sir. So another question was there, but I think it is already answered. The question was from Chirag Sengupta. HER2 is a kind of tyrosine kinase receptor. Yet, ask this question.
Yeah, it is a receptor tyrosine kinase. It belongs to the EGFR family. So if you go there, the EGFR family is there are four receptor tyrosine kinase in the EGFR family: EGFR, RB2, which is HER2, the protein level. There is RB3 and RB4. HER2 is very prevalent in breast cancer, whereas EGFR is very prevalent in lung cancer. So yes, a short answer is these are all receptor tyrosine kinase, and most of the receptor tyrosine kinase are targetable, and most of the drugs targeted delivery, targeted drugs being developed are against receptor tyrosine kinase.
Thank you. So another question from Monidas, although this question is a preliminary one, what are the stages of cancer and what do they mean?
So stages of cancer, broadly, any cancer can be, again, there can be a little difference, but cancer can be staged from stage 1 to 4. So stage 1 is the early stage, then stage 2, stage 3, stage 4 is the worst stage. Stage 4 is when the tumor has metastasized to other organ sites. So stage 4, if somebody has a lung cancer, it has metastasized to bone, let's say, or somebody has breast cancer, it has metastasized to bone. So stage 4 is the most aggressive form of cancer. So most of the stage 1 cancer, which is early stage, are curative, and they can be easily cured with surgery and maybe radiation after that. Most of the patients, if they're caught at that early stage, do very well with surgery. It becomes really tough. So chemotherapy and targeted therapy and all these type of drugs, systemic therapy is more relevant when the cancer has advanced significantly. They are either stage 3 or stage 4, where surgical removal is tough, because imagine if a cancer has spread from lung to bone, you cannot have surgery in all other, like all on bones and lung and all this place. So surgery is not an option at that stage. So that's when, your systemic therapy comes in. So that's the stage of cancer 1 to 4. 1 is less aggressive, easily treatable with surgery and radiation. And stage 4, 3 and 4 is more aggressive. 4 is the most aggressive. And that is when you treat them with drugs, because you cannot treat them with surgery.
Sir, one question, although this one I am combining with another, Onkur Shikdar, he has asked, what is the role of HER2 in cancer? So already we have answered in your deliberation. And second one, Somnath Shah, HER2 receptors are mainly related to breast cancer or any other types also?
So the answer is it is relevant in other cancer. So what happened in breast cancer when the IHC or the protein-based staining studies was, it was discovered in 1980s, and it was all protein-based. And HER2 is expressed highly in breast cancer, but not expressed highly in other cancer types. So the initial relevance was discovered in breast cancer. But with sequencing studies, people have started finding mutation in the gene in different cancer types. So now with sequencing, people have started finding that actually this gene has a role in many other cancer types than what was initially thought. So the short answer is yes, it is relevant to many cancer types, mainly solid tumors, not blood cancer types, mainly different solid tumors. And the relevance has come to the forefront with sequencing studies, when people have started identifying mutation in the gene. When they were just looking at protein expression, it was limited to breast cancer because it is not highly expressed in other cancer types, but is mutated in other cancer types.
Okay, thank you. Now, Mr. Somajit Panda, he has asked one question. Can we summarize different cancers to only some set of common traits?
So if by traits you mean, so there is the way classically cancer is classified is based on pathology, right? So you have your lung cancer, breast cancer, brain tumor. So the cancer is segregated based on the tumor site and the pathology of that tumor site. So that is the classical definition. But to answer the question now, there are molecular subtypes of cancer being developed. So for example, if we take the example of HER2 and RB2, because I've been presenting on that. So now if patient, a lung cancer patient, a breast cancer patient, or an ovarian cancer patient have been found to have HER2 mutation. And if HER2 is the main driver driving those cancers can be now molecularly classified as the same cancer type. So this is a new concept which is now coming to the forefront, where people are saying, can you have molecular classification of cancer? So you're not looking at the specific pathology or histology and classifying cancer by their organ site, but you're classifying cancer by their mutation or genomic profile. So any HER2 mutated cancer can be a same genomic profile, even though the organ sites are different. Any EGFR mutated cancer types can be classified in the same molecular subtype, even though pathologically they might be lung cancer, breast cancer, or other cancer. So I think there is this concept now in play where people are classifying, or scientists are classifying cancers based on their molecular profile and not based on the pathology or histology when they're identifying the main drivers for them.
So, sir, Samarjit Bose, he has asked how biomarkers are helping the drugs to find out the overexpression of receptor proteins.
So biomarker, the way it is working, right? Once you identify what is the key player or key gene, you want to test it either through protein expression analysis, which is mainly immunohistochemistry, or genomic. So let's say, there is a lab, a scientist finds out that protein X is being overexpressed in a particular cancer type, let's say in lung cancer, or it is highly mutated in lung cancer. Then once you publish it and you show strong data in mouse model that, okay, when you inhibit this biomarker or this protein, you inhibit growth, and there is 10% of cancer patients who have mutation in this gene. So now we have a strong biomarker, right? The scientist, this lab has developed, they have found protein X, which is mutated in 10% of lung cancer. When you inhibit this protein, you inhibit growth. So now pharma company will be very interested and say, okay, this protein X can be biomarker for this 10% of patient, and we can try and develop drug against it. And then the pharma will come in and try to inhibit that protein and develop drugs. So that's how I think biomarker gets developed. When a lab finds something, they saw strong clinical relevance that protein has in that cancer type, and you publish it, then you establish it's a biomarker. And then pharma companies will come in and try to develop drug and see if they can help those 10% of those patients.
Okay, sir, 3 more questions we have here. Once a cancer cell emerges, how long does it take to become clinically evident?
It depends cancer type to cancer type. Some cancers are very slow. For example, I work a lot on prostate cancer. It can have a very slow progression. Like a person can live with prostate cancer for 10 years. It can be very slow. And there are certain cancers which are very fast. They will come, you will probably have a diagnosis and it progresses very fast. So it depends on the cancer type. There is no one answer to it. It depends on the pathology. It depends on the cancer type and what characteristics that cancer has. Some might be slow growing, it's the nature. Some might be fast.
So Saubharta Mukherjee have asked, diethylstilbestrol is a form of estrogen that was given to some pregnant women in the US during pregnancy. Now, pregnancy have an increased risk of breast cancer. How this drug can lead to cancer? Is it the correct information that diethylstilbestrol will increase the risk of breast cancer?
There are studies. So what happens, these are estrogen stimulators. So it can induce the risk of ER positive breast cancer, but I have to personally do a literature search to, to give a confirmatory answer. But based on what, the student has come up with, it's a estrogen agonist. So if the person may have a tendency for ER positive breast cancer, or it may have a possibility of inducing the ER positive breast cancer. So there could be a possibility, but I have to read myself to answer that in a more correct manner.
So Kunal Datta asked, so is there any side effect of hormone-based cancer therapy?
There is side effect of all cancer therapy. Hormone-based cancer therapy will have side effects. Sometimes they have really strong side effects because you have hormonal regulation, and it leads to like massive side effects. So yeah, the answer is yes.
And one small question is, does the deficiency of any vitamin responsible for the development of cancer? Is there any information like that?
They might be risk factors, but I don't think lack of any vitamin can cause cancer. So the thing is, when you have, if somebody develops any tumor, then different things can exaggerate it. But I don't think there is any clear correlation that you have a lack of a certain vitamin and you develop cancer. So there are two aspects of it.
So thank you very much, sir. We have come to the end of the questions.
Oh, thank you very much. It was nice
just
talking to you guys.
So they are got very interested here. So sir, we have all the participants, we are ending the session. We have experienced an extraordinary learning session with Dr. Sunny Guin. He has explained the connection between genes and cancer in a very lucid manner. The oncogene and the tumor suppression genes, receptor, tyrosine kinase as oncogene, protein HER2, gene ERB2, and its role in breast cancer patients. Now, his first-hand experience of targeting the tumor cells by immunoliposomes, he shared his experience with us, how the scientists are using the precision oncology into our daily treatment of cancer patients. So thank you, sir. You have burned your midnight or night oil today. At least now it is, I think, about 1 a.m. to enlighten us with your knowledge and experience. Thank you again.
Oh, no.
You again for sharing with up-to-date information in the area of gene and cancer with us. So, sir, have a good day ahead. Thank you.
Thank you. Good. I really enjoyed talking to the students and taking questions. So there were a few questions. So hopefully I was able to engage with the students, and I hope this helped.
So you are not seeing our faculty members all in one screen?
Okay, yeah, I see them.
Yes. And students
okay,
are.
Thank you very much.
Thank you, sir. So all the participants, now I am ending the session. So now please go to our website. Their feedback form will be there. You just click on it. I am opening the response form. Okay, thank you all. Have a good day ahead.
Bye.
Bye.